very much, Mateus, for agreeing to join the transplant webinar series that we have ongoing, part of the cost action program called Transplant, which I'm sure you know, led by Federico and Daniele. So it's our pleasure today to have Mateus. This is a talk, as you see, entitled Liquid Biopsy for Pancreatic Cancers. We've had a number of liquid biopsy talks, which is quite important, early detection, etc. So we've had some great talks, and this will be a great way to round up the liquid biopsy part. So Mateus is a researcher and public health technologist at the Oswaldo Cruz Foundation in the Institute Carlos Chagas in Brazil. He's currently, correct me if I'm wrong, the coordinator of the Laboratory of Science and Applied Technologies in Health. He has a lot of research and participation in a lot of different consortiums. He was part of Pandora. As most of you know, he's also an expert and is involved in studies with circulating non-coding RNAs as diagnostic prognostic markers. He is a fantastic speaker, and I think everyone will enjoy his talk today. So, Mateus, without further ado, the floor is yours, and thank you again for agreeing to be part of our webinar series.
All right. Thank you, Bruno. Thank you, everybody, for the invitation, the opportunity to talk with you. When you first invited me to make the presentation, okay, yes, so that's a nice opportunity, but what would I talk, which the topic that should be something different. So I want here to show you something that I'm doing here. I put on the topic, on the theme of the talk, like liquid biopsy for pancreatic cancer, finding and challenge in the overseas scenarios. I just making a joke with this overseas because I'm not sure if I'm here the only person not in Europe, because it's a little bit far from you across the Atlantic. So we have very different scenarios, very different research context, money to do the research. So I want to give you an idea of what I'm doing, the results, and the challenging and the difficulties and the opportunities to make research here. As Bruno said, I just want to make a quick introduction about myself and what I'm doing here and where I'm from. I don't know if some of you knows Brazil, knows I'm from Curitiba, that it's a city here on the south of Brazil. We are in the state of Paraná. It's around 400 kilometers from São Paulo, the second red dot here. And we are very far from Amazonas. It's not close. And we are almost 2 million inhabitants here in Curitiba. We have the oldest university in Brazil. It's 1912. That is some kind of funny with Europe, because your oldest university is more than 1,000 years ago. So we have 100 years of university. We have two soccer teams here, Curitiba and Atlético Paranaense. They are not that good soccer team. We have no beach. We have a very weak carnival here. So we are not like Rio de Janeiro. We are a different city. We are in a very nice city. If you want to come to visit, it's a very nice place to be. And where do I work? I work in Fiocruz, that it's a public foundation, research foundation here in Brazil. It was created in 1900 by Oswaldo Cruz. It was the guy who discovered the Chagas disease, all the entire cycle of Chagas disease, since the insect bites until all the parasite cycles inside the host. And we are a federal institution allocated at the Ministry of Health, and we are historically dedicated to infectious disease like Chagas, malaria, leishmaniose, all this kind of disease. And we also are historically focused in basic sanitation and public health. But recently, in maybe 20 years, 15 years, we are moving to chronic disease like cancer, neuro disorders, cardiovascular disease, because we are also we are yet a country with a lot of infectious disease, but we need to make a slow migration to chronic pathologies. We Fiocruz also have a fabric and for drugs and vaccines. We are not just research; we also make productions for our public health system, and we are around to 4,300 employees, plus not fixed employees and students. So we have also the research and also the teaching with post-graduation programs. So Fiocruz, this one is the castle. There are no the main building is in Rio de Janeiro, and we have also more 5 regional units. One here in Paraná, another in Minas Gerais, 2 in the Northeast, in Bahia and Pernambuco, and 1 in Amazonia. And this is a picture of my institution here. And how I came here, just a very quick talk. I don't know, around to around 10 years ago, maybe more, maybe less, 8 years ago. I just contacted Federico by email because in my postdoc, my PhD, I worked with pancreatic cancer, in vitro assays, cellular biology of pancreatic cancer, microRNAs, KRAS. So I contacted Federico to make some collaborations, to make some studies, and I know I bothered him several times with an email to ask him to answer me, and we start collaborating, as Bruno said, with some GWAS experiments where I collect samples, pancreatic cancer samples here in Brazil. Okay, so I was a little bit insecure to talk about pancreatic cancer for all of you that you know everything about it. So I'm just trying to make a very small contextualization and why I'm studying to try to understand the rationale behind my idea, my project. So why I'm studying liquid biopsy in pancreatic adenocarcinoma. As I said, everybody knows that we have non-specific symptoms like dark urine, jaundice, abdominal pain, fatigue, and weight loss. And this is also correlated with the clinical and epidemiological concepts because on this context, we don't know which is cause and which is consequence because these non-specific symptoms drives to late diagnosis or they are due to late diagnosis, limited surgical procedures, ineffective therapy, and low survival rates. So these two contexts, they cross-talking, and we have a late diagnosis because we don't have specific symptoms, and when the patients have the diagnosis, the tumor is advanced, so it drives to an ineffective therapy. So this is a kind of clear scenario. So the diagnosis is mostly based in image testing. When the tumor is quite big, it's okay. It's very easy to see and to have a confident idea that this is a pancreatic cancer. And mostly of the time you need to make a tumor biopsy to confirm this cancer by pathologist. But sometimes when you have a small tumor, you cannot be sure if on the image test if it is a pancreatic cancer. So you cannot also have a tumor biopsy because as it's a small one, maybe you cannot have a quite high quality of material, high quality of cells, high amount of cells to make the pathologist to confirm pancreatic cancer. And also we have a very big lack of non-invasive biomarkers. We use clinically CA19-9, but it's a nice test mainly for disease and disease progression. It's a great one to follow the evolution, the prognosis of the patients, but it's not that sensitive and specific for diagnosis. So it's almost a complementary exam, but everybody is doing, but it's not something very effective, in fact. And it's a very, again, to contextualize my idea, it's very nice when we study the genetics of pancreatic cancer. We have few cases linked with hereditary, like few correlated genes, especially BRCA and TP53, around 5-10% of pancreatic cancer, they are somehow linked with familiar cancers. And this drives to a low percentage. So on the other side, it's obvious to think that mostly of the mutations, most of the genetic alterations in cancer, they are somatic. They are acquired during the patient life, following some risk factors like pancreatitis, diabetes, alcohol consumption, and smoking. Everything you already know. So this is something very interesting that I said to link the rationale behind my project. We follow four four genes that they have a high rate of mutation in pancreatic cancer. the oncogene KRAS and 3 tumor suppressor genes, TP53, CDKN2A, and SMAD4. So they have a very high percentage of mutation in pancreatic cancer. This is a very nice thing on the research context because it's something more assertive, something more logical for you to look. And they also have, for sure, didactic and theoretically kinetic during the oncogenic process, because KRAS is mainly the first mutation to appear in a pancreatic cancer. It's a drive mutation in when the tissue dries from normal to low grade degree. And after that, the second mutation that usually appears, it's on the tumor suppressor gene CDKN2A, which between in the end of low grade to high grade degree. And after that, when the tumor evolves, it appears mutations in TP53. And the last one, it's mutation in SMAD4 when the tumor is already consolidated and it's drive to more advanced stage, more to a metastatic process than in the beginning. So this logical this logical mutational progression in pancreatic cancer is something very, very nice to see, where we can use it to make the diagnosis and maybe somehow to make some correlation with the disease, the stage of the disease, the complexity of the tumor. So why we're studying liquid biopsy for pancreatic cancer? The main idea, as I said before, is to have an early diagnosis. As we have late diagnosis, is also correlated with low survival, low effective from chemotherapy. And so the main point is to get an early diagnosis. But here's the point. If we want an early diagnosis, we have a small tumor. We want a small tumor. But as I said, a small tumor, you cannot see it very clear on image testing. You cannot have a biopsy for that. But also for liquid biopsy, if we have a small tumor, we have low cell-free DNA released from this cell, these tumoral cells in the bloodstream. So it's a problem also correlated with the sensitivity. So for early diagnosis, our biggest issue, our biggest point to reach, it's a high sensitivity on the process. But also we can make a genetic characterization of the tumor using liquid biopsy, because especially driving to target therapy, recently there are non-target therapy for pancreatic cancer, but now there is two drugs driving on the KRAS G12C, like sotorasib and adagrasib. There are two drugs initially for lung cancer, but they are making clinical trials for pancreatic cancer. So we can make also the genetic characterization of this tumor using liquid biopsy, using cell-free DNA characterization. We don't need to look for the tissue to see if they have this mutation and also to have a target therapy. But it's also, again, correlated with sensitivity. We need some clinical trials to understand if we can match the cell-free context with the actually the tumor genetic context. So these two types of logical process to early diagnosis and genetic characterization, we are trying to see if it works here, and to see how can we use this it works in a Brazilian real world. As I said, we are in Brazil, a very big country, a very we have a difficult of logistic process, because, as I said, it's a big country. We need to take samples from everywhere. We took a long time to buy some reagents that come from USA, from Europe. And also, is it liquid biopsy cost effective? Again, here in Brazil scenario, and can we pay for that? So on research context, okay, we have a research project, a research budget, we make some cell-free DNA, a liquid biopsy for some patients, but can we afford here in our public health context, make to translate this to a real world? So my project, it was a very simple thing, whereas we have some PDAC cases, patients, we make the cell-free DNA extraction, quantification, the molecular detection using NGS and digital PCR. I'm going to explain a little bit later. And we make also the data analysis. So now just to try to understand a little bit better and to now starting to see like the points and difficulties that we found, maybe not just here in Brazil, everybody who works with liquid biopsy, that the first step is to have associated with sensitivity when we work with the plasma volume and characteristics. For example, we can extract the cell-free DNA from 1 ml, half ml of plasma until 15 ml of plasma. It depends on, especially on your budget and on your project. For example, I just bring here some points here. The kit that I'm using to make cell-free DNA extraction, it costs around 2,000 euros. If so, 2,000 euros if I can make 50 extractions using 2 ml of plasma. Okay, I want to have more plasma. I want to improve my quantity of cell-free DNA extracted. Okay, so I'm gonna use 5 ml. So I'm gonna make just 20 samples. If I use in 10 ml, I'm gonna make just 10 samples. It's a logical thing. So if I use more plasma, I'm gonna have more cell-free DNA. I'm gonna have more sensitivity, but my project's gonna be more expensive. So you need to think about your sensitivity and your money to spend on that. And also the plasma characteristics, it could be sometimes very jaundice that we have see that there's no big difference on a jaundice plasma on context on the cell-free quantification and extraction, but hemolyzed plasma, it doesn't work. All hemolyzed plasma we cannot use. We extract the cell-free DNA quantification, but it's not useful on the molecular detection. Another point here and another difficult point is about the initial processing, because you need to collect the blood and make a quick manipulation and centrifugation. We use a protocol that there are two centrifugations, two consecutive centrifugations, and we make an instant freezing at -80 to not degrade the cell-free DNA. So this is something easy when you do inside your lab, when you do with a great hospital. We have all the infrastructure people to do that. But maybe when you do in a small center, in a small city where you don't have this kind of thing, the people get the blood and let in on the freeze for, I don't know, a long time or period, some days, it all degrade the cell-free DNA. So this is a logistic process to sometimes complicated. And we also have the collection tube. For example, in this project, we use DTA tubes, but we have packaging and extract tubes. There are some tubes specialized for cell-free DNA collection. They make the stabilization, avoid the degradation of this material, but again, it costs more than a normal tube. For example, a packaging, it's around 15 euros a tube. So depends how money it should be better. It should be a nice approach to use packaging extract tubes. But if you have the money, if you don't, you need to arrange your project on that. And another difficult that I don't mean something difficult, but another interesting point on the cell-free DNA, it's about the quantification. You make you extract the cell-free DNA using any kit you want from your plasma. So you can make as a normal nucleic acid, a normal DNA. You have 3 options of quantification: spectrophotometry using nanodrop, fluorimetry qubits, and microfluidics. But as it's not easy, cell-free DNA, you cannot make the quantification using nanodrop or qubits. So are the easiest and the cheapest one. So you need to make the quantification using microfluidics, bioanalyzer, or tape stations platform. So it's something more sensitive. It's a very nice approach. It's a very good one, but again, it requires more money and it requires more infrastructure for make this quantification. And the last thing you can need another thing you need to think on your idea, on your project, it's about your molecular detection, your genetic detection on this cell-free. Mainly, you can use three different approaches, like next generation sequencing, real-time PCR, or digital PCR. That is, each of these are correlated with your project idea and again with your money. Because if you want to make a specific search for a specific hotspot mutation, a specific target mutation, a specific gene on your cell-free DNA, you can make using easily a real-time PCR, a digital PCR, or if you want to do something more broad, more explorative, using more, you need to make a next generation sequencing. And again, this is correlated with sensitivity and specificity. You need to think. Next slide, I'm gonna show you about this kind of sensitivity on the next generation sequencing. But another point here that we have to fill in on this, on the potential of translational liquid biopsy from the lab to a really a useful exam for some patients. It's about, you need to have a quick analysis. For example, if you have a pancreatic cancer, suspect some pancreatic cancer, you make a liquid biopsy for detection of KRAS. You cannot take 2-3 months to make the sequencing, to make the digital PCR, to return the result for this patient. If this patient wants to see if you have the KRAS G12C for using a target therapy, you cannot take again a long of time to return the result for this guy, because maybe it's going to be late. So your methodology, and if you are NGS, PCR, digital PCR, this should be correlated with your money and with your delivery time. If it's just a research project, you don't have this necessity of a quick response, but if you want, you need to adapt your system on that. And another thing important on the cell-free DNA analysis using NGS, it's, as I said, especially for early diagnosis, we need to look for a high sensitivity. But looking for a high sensitivity, we have mainly three variables on that. The input of our cell-free DNA, we're gonna make the library. The quantity of primers we're gonna make. I mean, I want to look for these 4 genes. I'm working here in my project. I want to look for 50 genes, 200 genes. This is gonna make also a cross-talking with your sensitivity and with your cost of your project, and also the sequencing coverage. In normal exomes, whole genome sequencing, you can have a coverage of 20, 50, 100x of coverage. But here in liquid biopsy, if you want a great sensitivity, you need to have a great sequencing coverage. For example, here, this is a quiet gene table, where they said if you want a mutant allele frequency of 0.5%, that it's very low, you need to have an input of cell-free DNA of around 30 nanograms and a mean read sequencing coverage of 24,000x. If you want to drop this sensitive variant allele frequency to 0.1%, you need to have 60 nanograms of cell-free DNA to start the library construction and 48,000x of coverage. I don't know familiarity of all of you with sequencing, but when we talk of 58,000x of coverage, it's a very, very big coverage. And when we talk of 60 nanograms of cell-free DNA, it's also a very big quantity of cell-free DNA. So this is a hypothetical thing. So if I want to have the best scenario to have 0.1 of sensitivity, I need to have a lot of cell-free. I need to have a very big read sequencing depth, but it's correlated with the sample. If you have this amount of nanograms of cell-free DNA, and with the money, how many samples you can use on your sequencing. I'm going to talk a little bit early on that. So my project, it was liquid biopsy for pancreatic cancer diagnosis by NGS through detection of mutations in KRAS, SMAD4, TP53, and CDKN2A in cell-free DNA. So it's not something very as a complex. So we got the cell-free DNA. So we got some PDAC patients. They came in from São Paulo Cancer Institute. As I said, we have logistical procedures here in Brazil that is not that easy. So we choose to collaborate with this institute that is closely to Curitiba, and it's the biggest oncological center here in Brazil. It's a public one, linked to São Paulo University, and they have patients treatment, surgeries, everything, and they have a research center and they have biobanks. So we collaborate with them. They collected the blood from these guys, pancreatic cancer patients that are enrolled on São Paulo Cancer Institute. We make the extraction of cell-free DNA of 5 mLs of plasma in EDTA tubes, make the cell-free DNA extraction using a magnet bead kit from Prigene, and make the sequencing for all just the exons. We skip the introns from all these 4 genes. And again, this project was a kind of my first project with liquid biopsy. It started around 3-4 years ago. So I never worked, our group never worked with liquid biopsy. So our inclusion criteria there was they were very soft. So I said, we want patient with pancreatic adenocarcinoma of pancreas. Okay, so it's in a stage 4 metastatic. Okay, we want this guy because clinically the liquid biopsy for this guy will not be relevant because okay, the diagnosis is also consolidate. It's a metastatic pancreatic cancer. There's no target therapy. It's not effective. So why do we want this guy to learn how to work with liquid biopsy? So this advanced metastatic pancreatic cancer, they are somehow like positive controls for our study to see these guys. We probably find a bigger amount of cell-free DNA. We probably will find more mutations on this cell-free DNA. And we also collect patients with or without treatment, because after that we're gonna make some clinical evaluations on the context. Our project was with a follow-up of patients for 2 years. That everybody knows here that it's somehow very challenging to follow up patients with pancreatic cancer for 2 years, because mostly of them died very fast, 6 months, 1 year, they are already dead. We have several blood collections according to initial stage and tumor progression. So we don't have just one sample collection of this guy. We try to collect multiple samples according to the return of these patients to the institute. It was around 4, 5, 6 months between one sample and the other one. And so we could follow the cell-free characteristic quantification and the molecular detection of the cell-free DNA with the clinical evaluation of this guy. So we have epidemiological, clinical, and treatment data associated. We tried to make association with FFP genotyping, like to say, okay, this guy, this patient, he have a mutation in KRAS on the tissue, on the somatic tissue, the tumoral tissue. So can we find this mutation on the blood? So this was something very challenging. There was a weak point on our work because we couldn't have the tissue from these guys, from any of these guys, because it's somehow very challenging to have this. We are trying to have this on the future. And we make a quick validation by dPCR, digital PCR, especially for KRAS. Okay, again, just to show you the logical of the idea, as I showed before, this progression of mutations, of appearance of mutations from KRAS to CDKN2A, TP53, and SMAD4, it was something logical to think, especially on the follow-up of these guys, as we have following up them for a while and have the blood in several points. So we make a publication on that recently this month on Scientific Reports on this idea. This is not a very, how can I say, it's an work to not focus on the molecular findings, not focus on the correlation of the cell-free genetic characterization with the patient evolution, with the patient progression. It was just the beginning of the story, where we want to talk what we're doing, what the difficulties we found here, what the preliminary results we found on this project. And we are doing another, finishing the project to have more samples, more sequencing samples, to make the correlation of the cell-free, of the genetic characterization on the cell-free with the clinical aspects. So on this project, on this paper, we had 55 patients, that counts 133 samples. We collect immediately and centrifugated twice, and we shared this plasma, 5 or 6 ml plasma, with frozen immediately at -80. We stored them in São Paulo on the biobank until they came to Curitiba, transport on dry ice, and we told them just for the cell-free DNA extraction. So the pre-analytical process, it was very controlled. It was very, very nice. Wow. Here is just a swimmer plot of our patients. As I said, we tried to collect some patients from the independent of the stage disease, of the diagnosis, also be correlated, also be defined if it's in treatment or not. So we have few patients with the first collection at the moment of diagnosis. Mostly of the patients, they already have the diagnosis, and as we can see, the black triangles, they are the blood collections. So we have a lot of a lot of patients that we just make one blood collection, and after that the patient died. We have some patients that we make more than, think we have a patient that we have 8 sample collections and survive. We are following this guy for more than 60 days, so we could have this time lapse for the collection of the patients. So we have the blood from the patients, all the samples, 55 patients, 130 samples. We make the cell-free DNA extraction using the beads kit, and we make the quantification of cell-free DNA extraction in the bioanalyzer system. So that's the first problem, the first issue on the idea, because, for example, this is a representative of the bioanalyzer results. We have the sample 2 and 3, a very strong band. We have a great yield of cell-free DNA, but the others, they are not that good. So the first point, the first problem here was the cell-free DNA yield for making the library for next generation sequencing. Our approach, for the library construction, it requires 10 nanograms of cell-free DNA in around 8.5 microliters of cell-free DNA. So we must have around 1.2 nanograms per ml of cell-free DNA to make a library. We tried to make this with 5 nanograms, but it doesn't work. It was not very good results. We skipped that, and we just say, okay, we need 10 nanograms of cell-free DNA. So mostly of the samples, we don't have this concentration, this yield of 1.2 nanograms per microliter to make directly the library. So what we do with these samples? Just throw into the trash and just do it with a few samples that we have this concentration? No, we tried to concentrate the samples. When we make the cell-free DNA extraction, we eluted them in 50 microliters and make the quantification. Okay, the quantification is low. We cannot make the library with this. We need to concentrate this guy. So we air dry this cell-free DNA using a speed pack and eluted them in 12 microliters to see if we can concentrate it enough to have the cell-free DNA quantity for making the library. So here's just a quick view. Overall, we have a concentration and succeed to have the minimum concentration required for the library construction in around 66% of the samples that we have not enough concentration before. So, for example, here in green, the left column there for before the concentration, the right column there after concentration. Those in green samples, there are samples that before we don't have enough concentration for make the sequencing, for make the library construction. On the right column we have, it was good, it works. And the red one, it not that great concentration, but we still try to make it. And the black ones, they doesn't work. Actually they work, they concentrate a little bit on the cell-free DNA, but not concentrated enough for making the library. So this first thing, we for here another visual thing to see. This left graph here shows this red line, horizontal line, show this is the threshold that we must have, 1.2 nanograms per microliter to make the library. So the samples should be above this line. So we can see just in here in the right side of the graph that we have this enough concentration. Mostly of them they are above. And here we have a representation of our samples. Again, the red line horizontal here is the threshold to have the minimum concentration. So here, all these samples, they are above concentration to make library. We make the concentration with the speed vac. It works for around, as I said, 230 of the samples. So this was our first finding, our first learning on that, mostly of the cell-free DNA, mostly of the PDC plasma. Will not have enough cell-free DNA for directly making the library construction. So we need to make this concentration. And after that, mostly of the samples we can use. So here it's the final idea. We have our final cohort. We have 130 samples from 55 patients. We're making the cell-free DNA extraction. Just 44 have enough concentration directly when they were eluted in 50 microliters, and the other 89, they're low concentration. We make the cell-free concentration in 82 of these 89, and 48 we reach enough concentration quantification, and 34 not. So we make a final sequencing in 92 samples, this 44 plus 48, and this 34 we didn't make the NGS library construction. We can use them for digital PCR, for any other studies. Our alternatives for that, we don't want to lose these 34 guys here. Our alternatives for that could be more plasma. If we use more than 5 mLs of plasma, if we use 10, 15 mLs of plasma, probably we're gonna have more cell-free DNA. But as I said, it comes with more costs. It's a more expensive project. We can use specific tubes, like as I said before, we can use Paxgene tubes to see if it's gonna be more concentration on the cell-free DNA, and also look for some alternative for extraction methods. I'm making something like this. I'm gonna show you on the last slide also. So after we make the cell-free DNA extraction, we make the concentration, we get those samples. Okay, these samples, we have the enough concentration to make the library. We make the library, and our sequencing was carried out in a Illumina MySeq V3 flow cell. That theoretically we can have 25 samples on this flow cell in an average coverage of 10,000x. So it's not, here we try to balance between a great coverage, 10,000x. It's a good one. It's not a sky high coverage, but we try to balance with 25 samples, that it's not a lot of samples, but that's not a few samples. And also with my money. For example, as I said before, this is also correlated with, if we use 50 samples here, we're gonna buy just one flow cell, but we're gonna have a coverage of 5,000x, not 10,000x. If you want a coverage higher than 10,000x, for example, if you want a coverage of 25x, we 25,000x, we're gonna just put 10 samples on that flow cell. So this kind of calculation, it's correlated with coverage, number of samples, the turnaround time you want to deliver to return this data, and the money you have. So the mean sequencing coverage for our samples here, it was around 6,200, with a range from 1,500 to 10, almost 11,000. So we have a very big variant of the coverage on these samples. But the average was 6,000. It was a nice one, but we want to improve that. And okay, we make the sequencing. We have for sequencing, we have sequencing all those samples. And after that, what's the point? Especially, I'm not a bioinformatician. I don't like black screens and commands. So we want to make this data analysis in a easy way, especially if you want to translate this for a system, for applied exam. We cannot take too long to analyze and to generate this data and to have the return for the patients, for the physicians. So I used a software from Quigene also. Its name is CLC. It's a very friendly and it's a very useful one to make it, but it costs money to make it. So basically, we import the sequencing from Illumina, data from base space from Illumina. We generated the sequencing, our sequencing pair sequencing by the pair reads. We make a quality control evaluation. We see if the sequencing reads the coverage was good, our target region. We say, oh, I want to sequencing the exon of these 4 genes. How was the coverage of these guys? We can make a quality control of also the sequencing and run the CLC analysis, which is a specific analysis for this panel of sequencing. It's a very short panel. When we talk in NGS, I was just looking for exon of 4 genes. It's a very, very small panel, and we can make this analysis individualized for this approach, this panel, and go to this all these arrows and these boxes and say which genes we have, our target region, our primers, and everything, and it can make the analysis in a very fast and straightforward way. And after that, this is just a representative of the result. For example, for one sample here, we have all the variants that he found on this sample. On the first column here in the both table, it's the chromosome and the type of the deletion, the reference, the allele. We have all the, when you're gonna run the sample, the analysis on CLC, you have a lot of some parameters, quality parameters you can give it to. So you can, these variants here, they are also filtered with the parameters you previously set up. So there should be confident. So here we have the most important things here. It's about the count and the coverage. The coverage, it's how many times that variant appears on the sequencing, and the count, it's how many times the mutant variants appear on the coverage, and it gives a simple division on that, gives your frequency of this variant, this mutant variant on the sequencing. It also have another point that say about the quality of the base quality of the sequencing, where it should be higher than 30. When you have a base quality of 40, 50, as much higher as it's better. So you can look at this and to see, as I said, especially the frequency of your variant and the quality of your sequencing. And here you can see that we have frequency since 0.71%, that we found 9 variants with substitution of from T with a C. 9 variants in 1,271 samples, until almost 100% of frequency. So this is another thing that we must to see. We need to look if we have frequency close to 100%. Probably, very probably, they are not somatic one. There are germline variants. This cell-free DNA was released from the tumor sample. Okay, could be, but also from normal cells. But so this is not correlated, probably not correlated with the tumor. And when you have a frequency also close to 50%, we somehow also correlated with an heterozygous variation, allele variation, and it's also not correlated with the tumor. So in this kind, we are looking for the frequency with a very low frequency percentage. So here, just a representative, how many variants we found for each sample. And when we filter these variants with frequency less than 50%, that we can say, okay, this variant, it's not a germline variant, it's not just a different polymorphism on this guy compared with the database that we used. When we see the variants, the black bars, they're total variants, the gray bars, there are variants with frequency less than 50%. It was all around half of them. So we divided these variants. We just want, as I said, those one with frequency less than 50%. And after that, we have several variants. As I'm looking at, showed here, we have 860 variants with a frequency less than 50%. So we have 860 somatic, probably somatic variants. So, and then what we're gonna do with this 860 somatic variants to see if it's a pathogenic one, a benign one, a VUS. We can get each one of these variants and search on ClinVar or VarSum to see if it's a pathogenic one, pathogenic-like variants, a benign, a VUS. But it's gonna take a life to do this. So we use Quaigene software that's named QCI, Clinical Insight. You just it's a very straightforward thing. You get your VCF, you get your variants that you found on the CLC, and just copy paste it here on the clinical, the QCI, and it's like a software that talks with databases, with scientific literature, and gives you if these variants, which of those variants, if you have pathogenic variants, if you have benign variants, you have VUS on this sample. So this is a logical process we want to follow because, as I said, we cannot make it manually, but we just have a short, actually it was a trial just to make using the QCI for just 10 samples. If you want to make it for all of ours 133 samples, we need to pay. It's not a cheap thing. So we make this QCI in just 10 samples. We choose here, for example, I'm showing you the QCI results for these 10 samples. We select the samples that most of the patients are in an advanced stage 4 metastatic one, because there are those patients that we have more chance to have a pathogenic variant in any of these 4 genes. And we also select some samples that we have a very big coverage to see if you have more chance again to find these pathogenic variants. These 10 patients we use it on QCI. We found 2 variants in KRAS, G12C, pathogenic variants. We are now just focused in pathogenic variants. We found one G12V and 2 G12D in 3 patients, and we have a pathogenic variant in TP53 and another 2 pathogenic variants in SMAD4. So from the 10 patients, we just have 3 of them with pathogenic variants. The first one with 3 pathogenic variants, KRAS, TP53, and SMAD4. The second one, we pathogenic variants in KRAS and SMAD4, and the third one just in KRAS. So, okay, we found this. We can see that we can make a sequencing. We can make a analysis somehow friendly way, a very easy way, and we can use the QCI to evaluate the meaning of these variants on the patient context. After that, we just make a manual search on the pathogenical variants in all the other patients that we have. So, okay, we use the QCI just in 10 samples. Okay, now how about the other 80 patients, 80 samples? Do we have more pathogenical variants over there, over those guys? We need to find this. So we got on CleanVar database. We look for pathogenic-like and pathogenical variants in KRAS, TP53, CDKN2, and SMAD4. We just came there, put KRAS. It gives a list of all pathogenical variants of KRAS. We manually screen these pathogenical variants in our results. So this did the same thing, the same way for all the 4 genes. And we make pathogenic KRAS in 4 samples, divided mainly in G12D, 2 patients with G12V, and 1 with G12R and Q61H. We have 4 pathogenic variants in TP53, 4 different ones in 4 different patients, and 2 pathogenic variants in SMAD4 in 3 patients, 2 of this one and 1 of this one. So finally, we found that this was our final list of pathogenic variants found in these patients with the mutation frequency. As we can see here, there are small mutation frequency. The biggest one, it was this KRAS G12D in these patients with almost 50%. It was a huge quantity of cell-free or mutated KRAS on the cell-free DNA. Probably it's a patient in a very advanced stage. So we have 13 variants in those 58 patients, that in 13 pathogenic variants in 58 samples. So when we look at, especially for KRAS, as I said before, KRAS is the primarily mutation in pancreatic cancer. When you look for these 9 patients with pathogenic variant in KRAS, 6 of them had just 1 sample and died closely after the sample collection. So 6 of these 9, we have pathogenic KRAS, and they died very fast. And they detected it was detected mutated pathogenic KRAS in the last 2 samples of the patients 5 to 9 with increasing frequency. So this guy, we have 2 samples, consecutively samples for detection of KRAS, where this frequency improves from 3% to 49%. And this patient died. So this was a nice thing, not for the patient, but for the study, that we can see we improve our cell-free, our KRAS frequency in this guy. The tumor was evaluating, the progression was not good, and this guy died. But on the other hand, in 2 samples, we found pathogenic KRAS with a frequency around 4 and 2.5%, but the following sample was negative. However, the patient died with cancer. So this was nothing uncommon, was unexpected. So why the pathogenic KRAS disappeared from one sample to the other and the guy died? No, it probably was a methodological bias, methodological issue. When you look for these 2 specific guys, one of them, when you have the pathogenic variant, the coverage of the sequencing was around 5,200, and the following sample, where the pathogenic KRAS variant disappeared, the coverage dropped to 1,300. Okay, so say, okay, maybe the variant was over there. We couldn't see because our coverage was much lower. But in the other patient, on the same scenario, the coverage, they were similar. So doesn't make sense for us to that pathogenic KRAS variant disappear from one sample to the other. These I want to just to talk, because KRAS was our mainly challenging here because everybody knows that 90%, 95% of pancreatic cancer have mutation in KRAS. So we expect to have a much higher rate of pathogenic KRAS detected in cell-free DNA. So we can see that this things when we found in one sample and not in the set, then the follow one, we can find that we have some methodological issues challenging to detect KRAS by next generation sequencing. Because of that, we try to validate these 8 of these 9 pathogenic KRAS detection with digital PCR to try to look for a logical way. If we extract cell-free DNA, we don't want to see TP53 mutation. We don't want to see SMAD4 mutation. We just want to look for KRAS mutation for have this patient diagnosis. So we make the digital PCR in parallel with the next generation sequencing. We have a very nice correlation. Just one sample that it was a allele frequency of 0.67% in NGS. We could not detect on the digital PCR. All of them we could detect, mostly with a very similar allele frequency in both methodologies, but some of them with a much higher frequency in one than the other. So upon a critical analysis of the variants according to the patient outcomes, none of the 3 patients who underwent surgery and showed any evidence of cancer at the end of follow-up harbor pathogenic variants. So we have 3 patients that underwent surgery and they were good, they were curated, they don't have a visible tumor, they don't have suspicious of tumor, and they didn't found any pathogenic variants. 4 patients who underwent surgery and were alive but still had cancer also showed no pathogenic variants. They underwent surgery, but they not that good surgery. They have the cancer again, but a small cancer, and we found no pathogenic variants on these guys. And this finding suggests that pathogenic variants were exclusively associated with patients whose cancer remained present and displayed an aggressive behavior. And another point, I'm almost
Mateus,
finished.
sorry to interrupt you, but for the sake of time and to have some questions, what we need to wrap up, if
Sorry?
for the sake of time, if for questions, I don't know how much longer you need for the presentation.
No, I need more 5 minutes.
Okay, perfect.
Okay, that's great. Okay. And my almost the final thing here, gonna be very quick. And so this was our idea for the characterization, and as I mean to make some very straightforward, a very assertive NGS sequencing for liquid biopsy, but we also can find new things on this context as we use a small panel, but it's somehow a broad panel. We don't expect find something that we found here, especially in SMAD4, CDKN2A. They're not genes like KRAS and TP53 with a lot of studies. So we focus especially in the VUS. So the VUS, they are variant with a no significance. So they're not, nobody say they are not pathogenic, but nobody say they are benign. No, it's a genetic alteration, but I'm not confident to say it's a good one or it's a bad one. So it's some there are alterations, there are variants that we can follow. For example, we find a lot of VUS on our study. And for example, here we have, especially in these 2 in KRAS, we have 2 VUS in CDKN2A and 1 in TP53. These variants we can using them in a database to updating if these VUS are coming to pathogenic variants or not. Maybe in 6 months, 1 month, 2 years, they could change the criteria. And we have another interesting finding that when we look for uncharacterized variants that they are new in literature, they're not new. You have some points of these variants, but nobody also said, oh, it's a VUS. It's a variant that it's not really classified. We have a lot of this variants here, and we looked in a very interesting one it was this variant 8 here located in SMAT4. So it located that this variant shows statistically significant difference with hazard ratio of more than 4 in survival curves, because this variant was present in 5 of 18 patients who died, but it was absent in all the 10 patients who survived. So it was a variant with a very low frequency, with allele frequency, but it was just found in the patients who died and not in the patients in those patients who survived. It doesn't appear. So it's just, we're gonna try to study a little bit more on this variant, but to see that we can find new things, we can find some nice correlations with progression, with treatment, on this using liquid biopsy. So okay, no. So my final slide here is, and what I'm doing now. We are finished this project with liquid biopsy with more pancreatic cancer patients, to try to correlate them with the clinical and the demographic aspects. But we are doing a project focused on using liquid biopsy for early diagnosis. So as I said, early diagnosis, you have a small tumor, you have a few cell-free DNA on the bloodstream. So we have patients with suspect of pancreatic cancer or diagnosis with a very initial stage. So again, at São Paulo Cancer Institute, it came a patient who is suspicious of pancreatic cancer. You make the imaging exams, and the physician say, I'm not sure, let's see if this guy, let's keep following up. So we collected around 30 mLs of blood from this guy in oxygen tubes, and they did this blood. It's transported by land, by trucks here to Curitiba immediately in 24 hours. And we extract the cell-free DNA on this guy from using 12 mLs of plasma. So we use more plasma than we used before because it's an early stage. So we want to have more sensitivity. And we make the cell-free DNA quantification and characterization. And if we have enough material to make a sequencing, great, we're gonna make. But the main focus here is to make digital PCR for KRAS. So we are gonna make a digital PCR for the 6 or 7 most hotspots, 6 or 7 hotspots for KRAS mutation, and to see if we can find KRAS mutated on this guy. And we are keep following up on the hospital. So we receive the material, make the cell-free DNA extraction, run the digital PCR for KRAS mutation, and okay, you don't find any KRAS mutation, and ask it for the hospital. Oh, how was this guy? It was pancreatic cancer. No, it was another thing. It pancreatitis, another kind of disease. And we're gonna correlate our finding here on the lab with the diagnosis of this guy. So this is a kind of scenario we want to see in a real world, because we are improving more logistical issues here. As I said, we using oxygen tubes that could be transported at the room temperature. We don't need that great manipulation on the lab to centrifuge to immediately froze at -80. So we try to have a more real world context here in Brazil, but we also insert more some financial issues as to use the oxygen tubes, and we are extracting cell-free DNA in of 12 mLs of plasma. It's going to be a more expensive study. So I finished from now, and just want to thank you everybody for the attention, for the opportunity to give you. This is our group. It's a kind of a small group here in Brazil. We work with cancer a lot, many types of cancer. We have a guy with proteomics, a guy with organ on a chip. We do a lot of things. We need to have a lot of projects to have money to work. So that's great. Thank you for attention, and feel free to questions.